Shalom, everybody. My name is Danny Michaelson, and I'm going to be talking about ApoE4 as a potential therapeutic target for Alzheimer's disease. The key issue in Alzheimer research, and indeed in any other biomedical research, is to identify the therapeutic target against which one wants to direct the research, because clearly, if we choose the wrong target, we will never get there. So what has been the field doing in this regard? The field has been choosing the therapeutic target based on several criteria. The recurrent approach is based on pathology of AD and on the genetics of familial AD, which is a very rare form of the disease. These studies have led to the discovery of important molecules such as beta-amyloid, presenilins, and tau. However, clinical attempts based on these molecules to develop a therapeutics to Alzheimer's clinical trials have been extremely disappointing. So the question that we have to ask ourselves now is, what is the take-home message from the lack of success of this approach? And in doing so, we want to focus our attention to the fact that Alzheimer's disease is very heterogeneous, as shown in this slide, and not all the patients show all the features, but the fact that it is such heterogeneity brings to light the statement that the possibility of developing a magic bullet that will be successful for all AD patients is very unlikely. Instead, what one needs to do is to focus on these defined subpopulations of AD, such as those defined by genetic risk factors, which have something in common and therefore would limit the field at which we are working. One such molecule is ApoE4, which is the topic of our presentation today. Now, what do we know about ApoE4? We know that ApoE4 is a lipoprotein, meaning it has a protein chain, polypeptide chain, which wraps some parts of it lipids. And it's well known that there are several isoforms of ApoE, which differ by the amino acids at positions 112 and 158. In particular, ApoE4 has two arginines there, and let's say ApoE3 has a cis and an arg. Now, the seminal work of Alan Roses, the late Alan Roses, has discovered the fact that there's a strong correlation between AD and the ApoE4 alleles. As you can see here, ApoE4 alleles, the frequency in controls is 26, whereas in the AD patients, it is 61%, which is rather significant. But not only is the prevalence high, because this may not necessarily indicate causality, but another important feature that was found by Alan Roses and his group was that age of onset is related to ApoE4. What we see over here is data of a few hundred patients, all of which were behaved normally at the age of 60. So they have, as shown over here, 100% clinically normal at the age of 60. And then as the age progressed, the disease started to appear. The important point is that if one is lucky to be ApoE4 zeros, no ApoE allele, you get the green curve, which shows us that about 50% of the patients become diseased at the age of 90, whereas if one is unlucky to have a homozygote for E4, this is the pink graph over here, you see that 50% of these patients become diseased at about 65, so 20 years difference. So this is very important. It's prevalent in AD. It has effect has a marked effect on the age of onset. If we look at the pathology, we see a lot of pathologies associated with ApoE4, which means that it does a lot of things, but it's problematic in the sense that we cannot tell which is a causality effect or just the downstream effect. But the effects are more plaque deposition in Alzheimer's, more inflammation, vascular impairment, and impaired plasticity and repair. Meaning, if one is ApoE4, one does not recover well from a repair type situation. So this is our ApoE4 molecule. We can say that it is worth studying, and it can be studied at three levels: the gene level, the protein level, and the downstream signaling level. I will start with 3, go up to 2, and at the end mention some preliminary results that we're doing at the level of the gene. Okay, so if we want to focus on downstream pathological effects of ApoE4, what we find is that there's a lot of phenotypes involved with ApoE4. And the ones shown over here is about 6 key issues. And this is, of course, very problematic because if we focus on the downstream and we choose to work on ApoE4 catabolism, for example, that it may end up that after 10 years of working on that, it is actually lipid metabolism, which is more important, or maybe a combination of these. This puts us in a very difficult situation to choose the downstream signaling as a guideline of how to work. And to illustrate this point, I want to address one specific problem, which is ApoE4 driven by a gain of toxic function or a loss of function? As we see over here, one possibility is that the apple is toxic. This would be a gain of toxic. And the other possibility is the slipper has been lost on the way down the stairs, which would be a loss of function. So we can ask the question, is ApoE4 mediated by a gain of toxic or by a loss of function? How do we address this question? One way to do that is to look at ApoE knockout mice and compare knockout to E4 mice. So if we have a function that behaves that it's not presented in ApoE knockouts, but it is presented in E4, we would call that a gain of function. So if we can see a few studies that were done with comparing E4s to knockouts, these are the position of A beta 42 and phospho-tau in the brain of the mice. You see that in both cases, the level of the pathological marker is markedly higher in the E4 than the ApoE knockouts, which are quite similar to the control. So looking at such data, one would tend to conclude that the effects of ApoE are mediated by a gain of toxic negative function. However, if you continue to look at other parameters, such as, for example, looking at synaptic-related parameters, we see that the picture emerging there is rather different. For example, if we look at the vesicular glutamate transporter, you see that the levels are lower in E4 than control, than E3, and that ApoE knockouts are the same as E4. So looking at this data, we can come to the conclusion that we cannot state categorically that ApoE4 is either a gain of toxic or a loss of function. And to us, a main take-home message from these findings is that we need to focus upstream on the ApoE4 molecule, on the structure of it, and not on what the functional consequences of these are, okay? Because as we saw earlier, 2 types of functional consequences, which are opposite in the interpretation, and therefore we think we have to focus on the molecule itself, what is different in the structure of the ApoE4 protein and the ApoE3 protein. So let's start in this direction. And the key point that has been observed in the literature is that ApoE3, shown here on the left, has more lipidation than ApoE4, which is shown here on the right. And the question is whether this is true and how important is that? And this is what I want to address in the next few slides. So we ask ourselves the question, we want to demonstrate that ApoE genotype affects the lipidation of ApoE. These are results from Yassin Hussein's lab, looking at the CSF of ApoE4 and non-ApoE4 carriers. And as you can see with the 2 lipids shown over here, phosphatidylcholine and sphingomyelin, the level of these proteins associated with lipoprotein in the CSF is much lower in the E44 for both of those lipids. So this is a clear illustration that the extent of lipidation is indeed lower in E4 carriers than in non-4 carriers. In addition to doing such measurements, which are actually ELISA measurements of the lipids and the protein associate, you can do what's called native ApoE gels. And native ApoE gels, what does is takes the material, CSF in this case, and runs it on a gel without SDS, without denaturing. So we're trying to get things separating by size in a native fashion. As you can see, when this is done with E3 carriers, you get the following pattern of 2 general populations, one shown over here and one over there. But the ApoE4 is missing the higher molecular weight population, which is taken to be the more lipidated, and heterozygotes have a mixture. So we have here illustration that indeed ApoE4 is hypolipidated relative to ApoE3. This is shown in patient CS, as I showed over here, patient CSF, but this is also apparent in animal models and targeted replacement mice of ApoE3 and ApoE4. So we have a phenomena here that ApoE4 is hypolipidated. The question is whether this is relevant or important in terms of the function of ApoE4 or the pathology driven by ApoE4. So in doing that, since we're talking about lipidation, we looked at the molecules which are involved in lipidation, and this is a new player in the game for us, ABCA1. And ABCA1 is a molecule that sits mainly in the plasma membrane, but also intracellularly. And what it's supposed to do, or what it does, it takes lipids from the outer leaflet of the membrane and transfers them from non-lipidated ApoE to lipidated ApoE, which, as you see, are those which have the circles, the opaque circles in them. So ABCA1 does this thing, and we wanted to ask the question whether, by playing with ABCA1, we can correct the hypolipidation of ApoE4, and whether this has any merit in terms of counteracting the pathology of ApoE4. So how do we do that? So we look at the biology of ABCA1, and you see one thing which is well known, that the promoter is an LXRE type promoter, and compounds have been discovered, such as bexaroten, which activate the transcription factors activation of the promoter and result in an increased level of ABCA1. This is very nice and can be used in the lab to ask the question that we're asking, but it has a limitation that in doing so, we upregulate not only ABCA1, but other NXR target genes, such as ABCG and ApoE. And therefore, it is necessary to employ a complementary approach using an ABCA1 agonist that does not change the level of ABCA1, but rather changes the activity. It's an agonist of this promoter. And one agonist has been developed by a company called Artery. It's a peptide derived from ApoE called CS6253. And this peptide has the ability in vitro to activate ABCA1 and get more activity related to it. So we decided to take both of these approaches and ask the question whether by promoting or increasing ABCA1 activity, either with an agonist or with a bexaroten, by doing that, we will affect the lipidation of ApoE4. We did both of those approaches. The results are quite published and are quite similar. So I'll go very briefly on the ABCA1 agonist type of experiments. So we treat the mice with this ABCA1 agonist, and we monitor the effects of the mice on total ApoE and then later on native ApoE. As you see with the total ApoE, the levels are higher in the E3 than the E4, that's well known, and the levels are not significantly affected by CS. So there's no effect on the levels. But if we look at the effect of the native ApoE gels, which represent the amount of lipidation, you can see that the control ApoE3 shown over here is markedly more intense than the control ApoE4 over here, showing that indeed in the mice E3 is E4 is hypolipidated relative to E3. When you treat these mice with CS, the important point is to focus over here, and what you can see is that the control becomes darker now, meaning that there's more lipidated ApoE. And this can be shown below in a quantitative way. You see that in the high and medium molecular weight domains of the gels, the levels in the E4 black are lower than the white E3. And when we treat with CS, we get a reversal of this effect such that the levels of the species of ApoE4 become similar to that of E3. Interestingly and reassuringly, the species which are least lipidated, the low molecular weight show over here, they are higher in the control ApoE4 than 3 because less lipidation, therefore more non-lipidated. And this effect is also counteracted by CS. So we have a tool or a way or means of changing the lipidation of ApoE in the mice. And now the key question to ask is, what have we done by doing that to the brain pathology associated with ApoE4? I will show only a few examples of that, as this has been published quite some time ago, several years ago. But as you can see, on the control level, the A-beta 42 there's an opaque band here, which is lacking in E3. Or if you look at the tau phosphorylation, there's a marked tau phosphorylation on E4, which is missing in E3. And these effects, which go with E4-driven tau in A-beta pathology, and these effects are counteracted by the treatment with CS, as you can see over here, and as you can see over here, and shown also in the graph, that the increased level of A-beta and tau shown in control E4s are diminished by treatment with CS. And this type of experiment can be done, has been done on several parameters, and I'm just giving you the overall conclusion. And the overall conclusion is that the ABCA1 agonist reverses the hyperlipidation, corrects cognitive impairments, which I did not show, but it's published some time ago, reverses tau phosphorylation and A-beta deposition, which we've seen, and also neuronal parameters such as decrease in V-GLUT and neurogenesis and changes of an ApoE receptor. So we have here a whole slew of parameters, all of them leading to the conclusion that if we modulate the system in a direction of activation of ABCA and increased lipidation, we can counteract the E4-driven phenotypes. As shown in below, the pathological effects of E4 are counteracted by reversal of the hyperlipidation of E4. Okay, so what do we do now? Since we have proof of principle that by activating ABCA1, we can correct ApoE4-driven pathologies, we wanted to develop a way that would allow us to find small molecular weight druggable ABCA1 agonists that could be used as potential drugs. So in order to find them or look for them, we developed a high throughput screening looking for druggable ABCA1 activators. This is shown over here, okay? So, and then we want to, once we have this high throughput screening, which is what starts with maybe 50,000 or 100,000 randomly selected compounds, you want to do a series of filtering to get down to the end product, which is fulfills the task that you're looking for and does that very specifically. So this means that we have to have a series of criteria be used as we filter down from the randomly selected. Now it's important to note that when one does screening, one can do phenotypic screening, where you measure if the compounds affected a bad phenotype, or you can do structure-based screening, which is whether the compound that you selected binds to whatever target you want it to bind to. And these two approaches, each of them have advantages and disadvantages. The phenotypic screening has the advantage that it is functional, but the disadvantage is difficult to improve potency of ligands because you don't have a target. On the other hand, if you do structure-based, the main thing that you have a structure-activity relationship, and you can use that to optimize your rates. So the conclusion is that we need to use both of these approaches in our study, and we would do so as you will see shortly. So what we do is after we start our high throughput screening, we're looking for drugs which the positive hits, meaning I would say that a minute determination positive hits that increase cholesterol reflux. We have a few sets of criteria that have to be fulfilled after one or to the other. So this is the first criteria. We have we screen our 50,000 compounds for their ability to activate the lipidation of a non-lipidated to lipidated ApoE. When we did that, we found about 12 or 14 compounds that gave activity in the range of our positive control, the CS peptide. I would say most of the compounds go between 50 and 150% of the standard, okay? So that was reassuring that we have some positive controls with results, not too many. So the next step was try and assess the hits by orthogonal assay. Now, this is very, very important because you want to show that the hits that you've selected for one criteria in increased cholesterol efflux can counteract the badness of ApoE4 by other criteria, and which is not related at all to your screen. And the criteria that we chose was plaque digestion. This is one takes brains of APP-PS mice, which have a lot of plaques, and one treats these sections, live sections, with astrocytes, where it is well known that ApoE3 astrocytes digest those plaques nicely, and ApoE4 do so less effectively. You see the plaques over here, and that's what we're going to look at. So here's a control section. Here's a section after treatment with ApoE3, and here's up to treatment with ApoE4. And when you quantitate, that's what you see. As I said, E3 digests nicely, E4 does not decrease the plaques, and what we're looking for are hits that reverse the inability of E4 to decrease the plaques, and this is called our orthogonal assay. When we did this thing to our hits, you see again, first of all, some hits like number 1 are completely inactive, so we drop that thing from our agenda, and the rest have a range of activities ranging, again, between 50 and 150% of our positive control. Next, we need to test the toxicity. That's trivial. And when we measure toxicity of the hits, we lost a few more compounds. You see those 1, 2, 3, 4 caused markedly strong effect on the cell death. So putting together these 3 criteria that are shown over here, what we have shown is functionally, we have functionally selected our hits. But the question now comes, what is the target of those hits? Do they really interact with ABCA1, or is it an indirect effect through some other known mechanism? So to do that, luckily, the structure of ABCA has been set down recently, or published recently, utilizing cryo-EM technology. And as it showed, the molecule of ABCA has 3 domains. It has an intracellular domain, which is an ATPase, has a cross-membrane domain, transmembrane domain 1 and 2, and has an externally oriented domain, which is composed of extracellular domain 1 and extracellular domain 2. So we rationalize that if our hits interact directly with ABCA1, it is a good point to start by looking at the extracellular domain, which are water soluble, so it made it easier to synthesize them and use them in assays. And as you can see, this is the assay that we use. It's called microscale theraphoresis, and its principle is shown here. And at B, you see a large molecule in yellow. You heat it with a laser, it warms up the area. As a result, the molecules diffuse away, and then once you stop, they come back. So this graph of the fusion of the large molecule is indicative of its features. And if the small molecule that you're studying affects the diffusion of the molecule, such as shown over here, either makes it stronger or weaker, that doesn't matter to us, but affects the diffusion, you can get a curve of showing concentration of ligand and how does the diffusion of the big molecule occur. So we did that and showed 2 examples of heat 3 and heat 4, and you can see very nice binding curves, which show that indeed, or suggest that indeed, these hits bind directly to ECD1. A very nice control are results with ECD2, where none of them compounds bound to it. So where we stand at the moment is that we have done high throughput screening, led to the discovery of 5 key molecules. And then when we looked at those molecules and focused on C7, which had the highest affinity and cholesterol efflux, and we did some biomedical studies utilizing available analogs, that and the results shown here are 2 such analogs that we received. And together with C7, that led to the idea that this could be an initial backbone. We continued to work around this backbone and found our best hit to date, which is N1, there's a KD4 micromolar, and we get a general scheme of the ligand that we're looking for with two down here, 2 rings and 2 R1 and R2 groups to be discovered or to be optimized. So once we hit this N1, we thought that we need to do some more studies with that to convince ourselves that indeed it is a good ApoE4, anti-ApoE4 molecule. So what we did is we looked at a few other phenotypes of ApoE4 that we have. For example, work of Mirna Safia in the lab has shown that the level of insulin receptors and LRP receptors are lower in E4 than E3. And interestingly enough, when we treat these cells with a positive hit, you see that the set levels of this receptor, insulin, were down in E4. And then when we treat with the drug, it goes back up to a level similar to that of E3, which by itself is not affected. With other receptors, the phenomena is not exactly the same, but the same principle holds, that E3 has more receptors than E4. And when we treat with the N1, we get the marked increase in receptor levels, which is associated with abolishment of the difference between E3 and E4. Another important thing which you wanted to know is whether it affects anything in the brain, because all the things that we've done so far were done in vitro. So we did some in vivo experiments. Dr. Alec was for Bar-El is the one who pursued them. And as you can see, when you inject N1 ICV directly into the brain, so before you inject it, you see the level of lipidated ApoE4 is less than E3, but after you lipidate it, this level comes back up to, but almost similar to the N3. See over here, and mainly in the high molecular weight species. So when you introduce the compound directly to the brain, it does what it's supposed to do. But of course, this is not patient-friendly, and what we need to do is to consider a way in which we introduce the drug in a more patient-friendly manner. And so the preliminary results that Alec did was intranasal administration of N1. And again, here you see, if you look at the quantitation, that in the high molecular weight and some in the medium molecular weight, the levels become higher in E4 after treatment. So we're optimistic that the drug can work when introduced peripherally and be active in the brain. One last point that I want to address, that these ABCA1 transporters play a role in other diseases as well. For example, in arteriosclerosis, it's well known that one of the pathologies is that macrophages accumulate a lot of lipids and become foam cells, which are pathological. And this is associated in many cases with downregulation of ABCA1. So if we have less ABCA1, less cholesterol efflux, more accumulation. That is well-known phenomena in arteriosclerosis, and it's been suggested in this field that activators of ABCA1 could be a way to reduce the foam cells, which are the carriers of the pathology. So Mariano Vadia in the lab, what she did was follow our previous cholesterol studies, but instead of working on astrocytes, which is what all our studies were, she worked on peripheral cells, murine foam cells, and macrophages. And as you see over here, that all of the drugs N1, CS, C7, CS, activate cholesterol efflux from both murine foam cells and human macrophages. So this has the potential of being used to address all sorts of atherosclerosis problems. So our very conclusion is that small druggable ABCA1 activates, which reverse the hyperlipidation pathological power of our E4, are being developed. Future plans are in two main directions. One is, of course, to try and get the drug to go from micromolar to nanomolar and to better characterize the in vivo concentrations. One last point, as you see, we talked here about downstream as not being very attractive as an approach, focusing on the molecule, which is our approach. There's also a possibility of focusing on the gene level, converting ApoE4 to ApoE3. And this is something which we have begun to work with Danny often in collaboration, and hopefully by next year we'll be able to show you some more results. So the bottom line of everything is ApoE tree of knowledge is ready to be harvested and translated. And of course, the group that has done the work is shown over here, and I want to particularly mention Dr. Alik Bar-El and Mayan Ovadia, who played a major role in the work on the small molecular weight cholesterol activated enzyme. Thank you very much.